Hi class, this is part 2. We're going over chapter 2. There are about 40-something slides in this set of slides. I'm not going to spend a lot of time on several of these because you'll get through some of these in your reading, and primarily we concentrate on chapter 1 out of these 2 chapters. But I do think it is important to touch on these. So chapter 2 starts on page 21 in your book. We're talking about the chemical basis of life. The box at the beginning of this chapter talks about how 71% of our planet's surface is covered by water, but then also the physiology of every organism, we're mostly water. So water is very important. So we look at the structure of water, atoms and atomic bonds, and then acids and bases. And one of your labs is based off of acids and bases this week, so we are going to cover some of this. So matter refers to anything that takes up space and has mass, can exist as a solid, liquid, or gas. It's composed of elements. So elements are substances that can't be broken down into another substance by ordinary chemical means. There's only 92 naturally occurring elements, and about 4 of them make up about 96% of the body weight of most living organisms. And you know these: carbon, hydrogen, oxygen, and nitrogen. Well, where do our elements come from? So normal chemical reactions don't produce these. The majority of the heavier elements, like iron, those are actually produced whenever a star explodes as a supernova. Those supernovas scatter the heavier elements into space, which then become parts of planets. And the iron in blood was actually formed from the explosion of stars. And I love this quote at the bottom: "After all, what nobler thought can one cherish than the universe lives within us all," by Neil deGrasse Tyson. So basically, we are made of the same stuff that stars are made of. You can find our elements, our basic elements, all throughout the universe. And it's a very humbling thought, I think. This figure is actually on page 22. It gives you a breakdown of the elements in living organisms. So we have a high percent of oxygen, 18% carbon, and then lower amounts of nitrogen, calcium, phosphorus, lesser elements like sulfur, and then some trace elements, and some hydrogen. So you can see the breakdown here. Now, atomic structure, we don't spend a ton of time on this. It's just the atomic theory states that elements consist of atoms. The atomic symbol, a name of an atom or an element, so you have like H for hydrogen, Na for sodium. These should be ringing some bells, maybe some chemistry bells. And then you have your subatomic elements like neutrons, protons, and electrons. Neutrons, they're neutral, there's no charge. Protons are positive, those are found in the nucleus. Electrons, they have a negative charge, and they're found outside of the nucleus, moving in orbitals. And then you have a mass number that's equal to the sum of the protons and the neutrons, and then electrons don't really have a mass. Atomic weight changes with gravity. You can take a look at this structure on page 23 if you like. I'm going to move past it. And I'll go over this really quickly, but all atoms of an element have the same number of protons. But that also gives the number of electrons if an atom is electrically neutral. So the atomic number, those are the facts on that. We know what the periodic table is. That is where elements, chemical, and physical characteristics recur in a predictable manner. Atoms are arranged in periods or rows, and groups are in columns. We've all seen the periodic table. This is a portion of that. I do want to go over the isotopes. So these are atoms of the same element that differ in the number of neutrons. This used to confuse me, but isotopes have, they're the same number of protons but a different number of neutrons. So they're going to have different mass numbers. Sometimes they're unstable, they might decay and emit radiation. Now, radioactive or radioactive isotope, that might be a word that you've heard before. Their behavior is essentially the same as a stable isotope of the same element. So you can kind of think of an isotope as a copy of another element, but slightly different. So not a twin, but very close. So these radioactive has a lot of negative connotation with it. Obviously, sometimes it's dangerous because it can cause damage to cells that lead to cancer, but it can also be used for benefits. It can be used to sterilize medical equipment or bring sterile food. but also it can be used as a tracer, like a PET stain. And this is an example of that. So the image on the left, so red is an indication where there are areas of greatest metabolic activity, and blue means areas of least activity. So on the left there is a normal brain, and on the right is from an individual with Alzheimer's disease. So we use radioactive isotopes for things like this. And so it can be beneficial. Now the picture on the top, this is a picture of the Fukushima nuclear facility. Following a tsunami, there was an accident here that released radioactive isotopes into the environment. So this was the dangerous kind. But then the picture on the bottom, radiation is being used to sterilize foods, like these eggs here, to protect against bacteria. So it's not all bad. They can be useful. And the stability of the isotopes, when it's said unstable or the half-life is different, that refers to the arrangement of electrons in their shell. So the arrangement of electrons in an atom, their electrons are always moving, so it's useful to make these models with energy levels, or like these electron shells, to kind of give you a visual. Each shell contains a certain number of electrons. For atoms up through the number 20. You have 2 that fill the first shell, 8 electrons fill each additional shell. And then for the arrangement in an atom, you have something called an octet rule for valence shell. That means the outside shell. If an atom has more than 2 shells, then the outer shell is the most stable with 8 electrons. When you don't have a balance of electrons, that's where you get the instability from. Atoms can give up, accept, or even share electrons to have 8. And then the chemical properties of those atoms, they're mostly determined by the arrangement of these electrons in the shells. And this is a really good pictorial example of that. So you have 6 of our important elements here. You can pause, take a look at this, or look at it on page 25 if you want. It really is just showing you how the more shells you have, the way that the electrons will balance themselves. You can stop and count if you want. Then we also have different types of chemical bonds. So a molecule, that's a group of those atoms that are bonded together. This here, you have O2, H2O, C6H12O6, that's sugar, and then nitrogen. And then you have a compound. That's a molecule that contains atoms of more than one element, just like C6H12O6 and H2O. So you have a compound. There's more than one thing going on there. It's not just oxygen, it's hydrogen and oxygen, or carbon, hydrogen, and oxygen, just a combo. Then you have 2 types of bonds. You have ionic and covalent. Ionic is the attraction between opposite charges, and then covalent is where they come together. They share electrons to complete the outer shell. So ionic was where opposites attract. This is where we get into the net charges and things like that. This is more like chemistry. We don't really stay on this for too long, but with ionic bonding, that's when 2 atoms are held together by the attraction between opposite charges. So for example, sodium has 1 electron in the valence shell, usually gives 1 up. So when you have sodium and chlorine that come together, chlorine only has 7 electrons in its outer shell, and so whenever they come together, it usually accepts a share on an electron from another atom. So the sodium has 1 more proton, and the chlorine has 1 more electron, so then they combine. And ionic compounds are often called salts. Here's a pretty good pictorial representation. That's on page 26 to show you how it forms together. And then with covalent, this is where 2 atoms are sharing their electrons. So 2 hydrogen atoms can share electrons to fill their outer shell, and then their orbitals overlap. As you can see in the middle, it kind of makes me think of a Venn diagram. The structural formula uses straight lines as a connection. So one line shows that you have a shared pair of electrons. But the molecular formula just shows the number of atoms involved, to keep it simple. You can also have double covalent bonds. That's where 2 atoms share 4 electrons, and double bonds this is really the main thing that you get off of this slide, is that you can have a double bond. You can even have a triple bond. But double bonds are stronger than single bonds, so they're harder to break if you need to break them for any reason. I'll move on past this. You can actually look at that on page 27 if you'd like. This again is another chemistry review. So you have a chemical formula and the reactions, and the way this is written is you usually have the reactants. Those are molecules that participate in reactions. Those are shown to the left of the arrow, kind of your starting point. And then your products, those are formed by reactions, and those are shown to the right of the arrow. So if you see a chemical formula, it'll usually have something written, an arrow, and then the reaction on the other side. Whenever we get into photosynthesis, you will see it's kind of like an equation. It's like 1 + 1 = 2. You've got an equation, a start point, and an end point. So we'll get into that a little later. But this is basically what it looks like. So if you combine carbon dioxide with 6 water, you end up with glucose and oxygen. In chemistry, you have to balance these equations. If something is unequal, you have to figure out how to do that. We won't work on that in this class, but it is good to at least brush up on the knowledge. For any of you that may be going on to chemistry, this will be a good, not really a starting point, but just something to know as you move on. Now, the real meat of this chapter is water's importance to life. So, life began in water. It is the most important molecule on Earth. We couldn't survive without it. All organisms are 70 to 90% water, and water has very unique properties, and that's why it's a life-supporting substance. And there's a lot of properties that stem from the structure of the water molecule. So it is what they call polar covalent, which means the atoms don't share their electrons equally. Oxygen is electronegative, or more electronegative than hydrogen. So electrons spend more time around the oxygen nucleus than they do the hydrogen nucleus. Oxygen, the end where the oxygen is, becomes slightly negative, and the hydrogens become slightly positive. So it's not an ionic bond or ions. It's a little different. It's just a little different. So the hydrogen bond in water, slightly positive of one water molecule, is attracted to the slightly negative oxygen in another water molecule. So in this case, they're kind of attracted to each other, if that makes sense. This shows you the structure of water. It's very important, but because of the structure of water, that's why things can dissolve in it so well. Because it's kind of like in between positive and negative, so it can attach and work with and dissolve a lot of things. So there are several different properties of water. I do ask you a question about this, so you can find this here in the slide, or you can read it in the book. Whichever way you look at it, it's up to you. So you have solvency, it can dissolve things. You have cohesion and adhesion, high surface tension, high heat capacity, high heat of vaporization, and it can vary in density, like ice versus water. So we use terms in biology like solvent and solute. So water is the solvent. It's the thing that does the dissolving. The solute is the thing that goes into the solvent. Like, for example, Epsom salt would be the solute that goes into water as a solvent. Due to polarity and hydrogen bonding, like I was saying, water can dissolve a lot of different things. Hydrophilic molecules are attracted to water, and hydrophobic molecules are not attracted to water. And so if you look here at the picture, this salt, sodium chloride, water causes it to break apart because of the way the positive and negative are attracted to each other. It's a very simple act, but the chemical way that it happens, I think, is interesting. So cohesion, that's the ability of water to cling to other water molecules because of the hydrogen bonds. So because of cohesion, water can exist as a liquid under ordinary conditions of temperature and pressure. And because there's such a strong cohesion, water flows freely but the molecules don't separate from each other. And then you have adhesion. That is the ability of water molecules to cling to other polar surfaces. So that allows water to be an excellent transport system inside and outside of living organisms. And that's how plants actually transport their water. They use adhesion. Here's a pretty good visual example of that. So in the roots or trunks of a tree, water is using well, first it's the cohesion of the water to stick to itself, and then adhesion for it to stick to the surface of whatever it's trying to move up, and it kind of works like a straw. It pulls it up through all of the pieces of the plant so it can reap the benefits. But another cool way to think about this is the picture on the right where it's a little bit more purplish colored. Our veins do the same thing. Water gets pulled through our veins in the same way. While we drink it, it gets absorbed and just kind of like travels because of adhesion. You guys have heard of surface tension before. I remember when I was little doing an experiment like this in science class where you would put a penny on top of water or a needle or something like that and just watch how it didn't break the surface. So the water molecules at the surface, they cling more tightly to each other than they do the air above, and that's because of the hydrogen bonding. And so because of that, you get this tension across the top of the water where you can set things on top of it, where you can see little animals or insects kind of skittering across it, usually really lightweight, something really lightweight. It has a very, very high heat capacity. So all of the hydrogen bonds that make those water molecules link allow water to absorb heat without really changing its temperature that greatly. So the temperature of water rises and falls really slowly. So for example, yesterday morning was 9 degrees outside. We had a low of maybe 4 degrees that night, but my pool water was about 32 degrees instead of 9 degrees. So it just, and I know this because I did a cold plunge. I jumped into it. Yes, I am crazy. Well, not crazy. There's actually a lot of benefits to it. But I know that because I checked my temperature gauge before I jumped in, and I was expecting it to be a lot colder. I thought it'd maybe be in the 20s, but water really holds on to heat. So sometimes water in lakes don't freeze even when it's cold outside because it takes a long time for things to cool down. Heat evaporization, that takes a great deal of energy to break the hydrogen bonds for evaporation, and then the heat is dispelled as water evaporates, like boiling water. Here's just examples of the heat capacity and water vaporization. Move on past that. I mentioned this as well. One of the other properties is varying density, because ice is actually less dense than water, which is different than other substances. So unlike other things, water expands as it freezes, and then ice will float instead of sink. And that makes life possible in water because ice acts as an insulator. Now this picture is on page 22. So whenever water freezes and you take a look at it, you have this very geometric design. So it has this pattern. And because of the way that they freeze in this spread apart pattern, that is what allows it to float. That makes it less dense because the molecules are not closer together anymore. And now we have the last part of our lesson. We have acids and bases. So water will dissociate into an equal number of hydrogen ions and hydroxide ions. You can see how it breaks apart in the chemical formula. So this is how we come into things that might be acidic. You have high hydrogen concentration. Whenever something is acidic, you have a lot of hydrogen. Those are things like lemon juice, vinegar, and coffee, many other things. And that's because the acids release hydrogen ions or take up hydroxide ions. And this is how we get acids. For basic solutions, that's where you have low hydrogen concentration, so things like ammonia or milk of magnesia. I think a basic solution can be more damaging in a lot of ways than acidic solutions. But they either take up the hydrogen ions or they release the hydroxide ions, and you can see that in the chemical formula below. And this is how we've come up with our pH and our pH scale. So the mathematical way to indicate the number of hydrogen ions in a solution is where the pH scale comes in. So it ranges from 0 to 14. 7 is your middleman. If it's below 7, it's acidic because there's more hydrogen. If it's above 7, it's basic because it has more OH. And then if it's 7, it's neutral. And here is your pH scale. This one's on page 34. So it kind of gives you a list of things. So things that are acidic, obviously we know hydrochloric acid is, but stomach acid, lemon juice, Coca-Cola, beer and vinegar, tomatoes or black coffee, and even normal rainwater is just a little bit acidic. Urine is also acidic. Things that are neutral, though, you have saliva, water, tears, blood is neutral. But then we'll move on to the basic side of things. You have seawater, that's a little bit more alkaline. Baking soda and stomach antacids. Great Salt Lake, milk of magnesia, ammonia. You have the bicarbonate of soda, oven cleaner, and sodium hydroxide. So these are just some of the things. But then we also have buffers. So there's chemicals or combinations of chemicals that keep pH within normal limits. So using a buffer, you can resist pH change by taking up the extra hydrogen or the extra OH. So the pH of the blood is about 7.35 to 7.45, but it's maintained by a buffer because we eat things that are acidic, and then we eat things that are basic. And we're fluctuating inside of our bodies all the time, and without a buffer, we would die. So the buffer helps maintain homeostasis within our body of pH balance. Then you have some diseases like congestive heart failure and diabetes that can result in what they call acidosis. That means the body can't buffer out those extra hydrogen ions, and if you don't treat that, then you could die. So it's all very important to be balanced. And that is the end of this lesson. So I tried to keep this one kind of short. I'll stop this here and let me know if you have any questions. Feel free to email me any questions or clarifications that you might need.